So for this semi-conservative mode of replication, we are going to have an experiment. That experiment has been done by Messelson and Stahl, and that is nitrogen isotopes. They have used nitrogen 14 and nitrogen 15, and with the help of that one, we came to a conclusion that DNA replication is semi-conservative. Semi-conservative means half of the DNA, means one strand of DNA is going to be coming from the parental DNA, and one half is going to be synthesized new one through a sequential events, that is one nucleotide every time is going to be added. That is the process, that is the semi-conservative mode of replication. Later on, when we are going to see the replication process, transcription process, and translation process, whole process you can divide in three broad area, three broad phases. That phase we are going to call it initiation, elongation, and termination. Initiation is just a kind of way how it is going to start the process. So it is assembly of all the initiated molecules, enzymes, that is called as initiation process. Elongation, just a kind of chain reaction, it is continuously going on. And last one will be termination, how we are going to end it and when we are going to end it, that is called as termination process. So replication process also we have initiation, elongation, and termination. We will see one by one. So what I have told you, in this case of DNA replication, in case of prokaryotic system, basic mechanism is same. In case of bacteria also, in eukaryotic cells also. But the major difference is in case of bacterial cells, because their DNA is very small, so only one replication bubble is going to form. Only one origin of replication will be there. Whereas in case of eukaryotic system, in our cells, we are having our DNA is very long, so we are going to start a replication at multiple places. So in our case, we are having multiple origin of replication. So replication bubble will form many places. So if you will see the complete DNA or longer form of DNA, there it will looks like beaded structure like this one. So multiple replication bubble, you can see it in our case. So this is the process when it is going to start. So replication starts at a specific place, both in prokaryotes as well as in eukaryotes, that is called as origin of replication. So there first helicase enzyme comes and it unwind the DNA. So DNA is going to be unwinded. Unwinded means both strands need to be separated. That is ultimately going to form a bubble-like structure. So helicase will open the hydrolyze the hydrogen bond and DNA will be unwinded. Then supercoiling is going to happen and that will be removed by topoisomerase. That exactly not part of the replication process, topoisomerase. That is just to remove supercoiling. This helicase is going to remove the hydrogen bond and we are having this type of bond bubble forms. Then comes second component which comes to this process is primer formation by a specific enzyme, that is called as primage enzyme, or you can call it chemically, it is technically it is RNA polymerase, because DNA polymerase cannot start from zero, cannot start from scratch. So it needs some previously existing, पहले से कुछ होना चाहिए था उसके पास, तभी DNA polymerase can build DNA. So सबसे पहले a primage enzyme will come and this will form a small RNA primer on both the strands. Both the strands means I'm talking about both the template. In case of DNA replication, both strands work as a template. Once we are going to have the primer formation occurs, then DNA polymerase comes, depending on which type of sequence we need to form. So either it will be DNA polymerase 3 for longer DNA formation, for a smaller DNA, it is DNA polymerase 1. As you can see, this is the half portion. If you will see, if you will consider this portion, so there, this side it is continuously going on. As unwinding will occur little bit more, then also this chain is continuously going on. But on this strand, it is slowly going to occur in a small fragment, and that is called as Okazaki fragment. This is just to maintain the polarity. Polarity means the orientation. 5 prime to 3 prime direction need to be maintained. That's why on one strand, so we are having continuous DNA formation, that is called a leading strand, and second one is lagging strands. Once this will form, then we are going to have a ligase reaction. Ligase normally going to replace the means the DNA polymerase which forms here. This DNA polymerase, it removes the RNA portion and replaced by DNA, and then ligase joins it, seals the gap, fill the gap. So you will have a continuous DNA strands like this one, and ultimately we will have two DNA strands. So this is parental strands which comes from here. This is another parental strands which comes from here, and this is the newly formed DNA. So this is called as semi-conservative. We have started from one DNA molecules which is a two strands. Ultimately we form two strands where half 50% is new strands, 50% is old strand, and this is the basic summary of DNA replication process. So the process which we have discussed till now is, first step is unzip the DNA molecules, unzip the DNA double helix. This is carried out by the helicase enzymes, which breaks the hydrogen bond. The separation of two strands forms a Y-shaped fork, that is called as replication fork. If two replication fork, two Y will be joined together, it will give you a replication bubble like this one. So I am talking here only half of the portion. This one, this is Y-shaped structure, that is called as replication fork, and complete thing is called as replication bubble. Both strands will act as a template. Remember that day first on your my first class or second class, I have told you, when you are going to do the protein synthesis, that time only one strand is going to be give you protein in our case, and that is called as non-coding strand, right? That is called as template strand or antisense strand. But when we are doing replication, that time both the DNA strands is going to be used as template. So this is one of the difference between the transcription and the replication. In transcription, only one DNA strand is going to be used. In replication, both of the strands are going to be used as a template. But on both the strands, direction is 5 prime to 3 prime only. So this direction is always maintained for all the four types of polymerases. Either it is DNA polymerase or RNA polymerase. Either it is DNA dependent DNA polymerase, DNA dependent RNA polymerase, or RNA dependent DNA polymerase, or RNA dependent RNA polymerase. Means all the four cases, this direction need to be maintained. So every time a new nucleotide will be added, and that will be only at the 3 prime end. So if to maintain this direction, on one strand, so we are having continuous DNA synthesis, long stretch of DNA synthesis, that is called as leading strand. And on another strands, we are going to have a small stretches, a small fragment of DNA synthesis, that is called as lagging strands. I will show you one video also how it is going to form. As a result of this, two strands replicate differently. Okay, so a new DNA strand is always synthesized in 5 to 3 direction. That already I have told you. Means this free 3 prime OH is a point where DNA is going to be elongated, where a new nucleotide comes. Because the two DNA strands are anti-parallel, the strand serving as a template is read into the 3 to 5 direction. How it will happen like this one? See, this is my DNA, and this is a small DNA strand has been unwinded in this case. So this is 3 prime, this will be 5 prime, this is 3 prime, this is 5 prime. So you know that DNA synthesis is going to occur in 5 prime to 3 prime direction. So if DNA synthesis will occur here, start from here, so this will be 5 prime, this will be 3 prime. So this direction it is going on. On the opposite strands, in this strands, in which direction DNA should be synthesized? In this direction, correct?
Yes, sir.
Fine. So till now here, up to here, DNA strand has been opened. Now little bit more DNA strand has been opened. So same, further it has gone, and more stretch of DNA has been unwinded. So this, on this strands, upper strands, this is continuously going on in this direction. But on this strands, because we have started from here, so this stretch is going in this direction. So for this portion, I need to start again DNA synthesis, and again it has to form this stretch. So on one strand, you are seeing continuously it is going on. As DNA will be unwinded little bit more up to here, again this will continuously synthesize. It is not going to stop at all. But on this strands, a small fragment of DNA is going to be synthesized. So this is called as leading strand, and this is called as lagging strands. Why it needs to occur? Because we have to maintain the direction of synthesis into 5 to 3 prime direction. This is clear?
Yes, sir.
And that is called as Okazaki fragments. This was first resolved. It was first discovered by Reiji Okazaki in 1960s, and because to give him respect, we are going to call it Okazaki fragments only. So what is Okazaki fragments? It is newly synthesized DNA fragment that are formed on the lagging strands, discontinuous strand during the DNA replication. They are complementary to the lagging template strands. The length of this Okazaki fragment in case of E. coli, so this should be italics. Whenever we are writing microorganisms names, any organisms names, it should be either underline, it should be italics. So sorry for this one. E. coli name should be E. coli. In case of E. coli, the Okazaki fragment's length is roughly 1000 to 2000 nucleotide long, whereas in case of eukaryotes, its length is very small. So it is 150 to 200 base pair long only. So the and in, if you will see the Okazaki fragment, if you just here, this Okazaki fragment here, RNA polymerase is there. So this portion is RNA polymerase. This portion is RNA polymerase. Here RNA polymerase is there. So two Okazaki fragments are separated by a 10 to 15 nucleotides RNA primer. That is written here. Both all the Okazaki fragments are separated by 10 to 15 nucleotides RNA primer, which ultimately later on it will be replaced by DNA molecules by DNA polymerase I. That also I will show it to you. So here this is RNA polymerase, this is RNA polymerase, these are RNA polymerase. Later on, like see, this DNA synthesis is occurs by which DNA enzyme, which DNA polymerase synthesizes this Okazaki fragment? DNA polymerase I. This is synthesized by DNA polymerase I. This is synthesized by DNA polymerase I. And this one is synthesized by DNA polymerase III. Clear? Now, in case of this one, DNA polymerase I came here. So it added nucleotide. After that one, it cannot go further without removing this RNA primer. So if DNA polymerase need to move further, first it has to remove this RNA polymerase. And the direction of this nitrogen nucleotide is, this is 5 prime and this is 3 prime. So first this DNA polymerase which is here for this Okazaki fragment need to remove one nucleotide from here, from RNA molecules in this direction. So RNA polymerase here requires, DNA polymerase here requires 5 prime 3 prime exonuclease. Normally we are having two types of enzymes. When in those enzymes which degrades your nucleic acid, either DNA or RNA, is called as nuclease enzyme. So those enzymes which digest nucleic acid are called as nuclease. These nuclease we are having DNase and RNase. Those which works on DNA called as DNase. Those which works on RNA called as RNase. This nuclease we are having two types. One is called as endonuclease and second one is called as exonuclease. Endonuclease and exonuclease. If you are having a DNA strand Like this is my DNA strand, and one enzyme which breaks the nucleotide DNA from the center in the between inside the strands, not from the terminal end. So any enzyme which digests your DNA inside the nucleotides, not either this end or not from this end. So that they are called as endonuclease, means inside the DNA. And those enzymes which digest your DNA from the terminal end, either from this side or from this side, they are called as exonuclease. This is clear?
Yes, sir.
When we are talking about exonuclease, so this DNA we are having, this is 5 prime and this is 3 prime direction. So if you are having one exonuclease which removes one nucleotide from this end, from 3 prime end, and then it removes another nucleotide from 3 prime end. So the direction of this exonuclease is 3 prime towards 5 prime. So we are going to call it, this enzyme is having 3 prime to 5 prime exonuclease. Those exonuclease which removes nucleotide from 5 prime end and moving towards 3 prime, for that one we are going to call, this is having 5 prime to 3 prime exonuclease. So exonuclease we are having two types, either 3 prime towards 5 prime like in this case, or 5 prime towards 3 prime. Clear?
Yes, sir.
Okay. Now when we were talking about one proofreading power. So see, in this case DNA polymerase 3, one nucleotide had been added here. For time being, this is the nucleotide which has been added, this is adenine which has been added. So DNA polymerase will go back and check this nucleotide is correct, right or wrong. Unfortunately this is a wrong nucleotide. So what will happen? DNA polymerase has to go back in this direction and remove this nucleotide. So what will be the direction of exonuclease? It is going to remove one nucleotide from this end, that is 3 prime, and towards 5 prime. So which type of exonuclease is proofreading power? Proofreading power means it is 3 prime towards 5 prime. So whenever we are talking any DNA polymerase is having proofreading power, means they should have 3 prime to 5 prime exonuclease power, capacity. Why? You have seen just now. Okay, wrong nucleotide has been added here at the 3 prime end. So DNA polymerase should look back and see it is right or wrong. If it will be wrong, they have to remove this nucleotide. So DNA nucleotide will be removed from which end? That is from 3 prime end. So direction will be 3 prime towards 5 prime. So it is called as 3 prime to 5 prime exonuclease activity. So proofreading means 3 prime 5 prime exonuclease activity. Clear?
Yes, sir.
So all the DNA polymerase, roughly, most importantly, bacteria is having 3 DNA polymerase: polymerase 1, polymerase 2, polymerase 3. Otherwise, if you see the details, total 5 DNA polymerases are there. Polymerase 4 and polymerase 5, they have been discovered at the in the roughly 1999, I think. So most of the books does not cover those 4 and 5. Most of the book contains 1, 2, 3 only. So all the 3 enzymes have this proofreading capacity, 3 prime to 5 prime. Every DNA polymerase is having 5 prime to 3 prime exonuclease, polymerase activity. They can add a nucleotide into the 3 prime direction, in this direction. Every DNA polymerase is having 3 prime to 5 prime exonuclease, means proofreading capacity. So all 3 enzymes, it is having this one. Now, this is two things, two capacity we have seen here. Third capacity which we are talking here is another exonuclease, that is called as 5 prime to 3 prime exonuclease. DNA polymerase 2 and 3 does not have this power. They cannot remove nucleotide from the 5 prime end. So here DNA polymerase 3 was there, it cannot remove any nucleotide from this end because they don't need it. Whereas if you see the DNA polymerase 1, this needs this 5 prime to 3 prime exonuclease activity. Why it needs? Because it has to remove one nucleotide from this 5 prime end. That RNA polymerase primer need to be removed. So DNA polymerase removes one nucleotide from here and then add a DNA nucleotide, means deoxynucleotides. Then remove second nucleotide and add second deoxyribonucleotide. So DNA polymerase 1 is having all three capacity. DNA polymerase 2 have first two capacity, and DNA polymerase 3 also have first two capacity only. This 5-3 only polymerase 1 is having. Later on I will give you table, but just try to understand. Any confusion in this one?
No, sir.
For others also I want to ask. If anyone is having any issue, you can ask me. For others also I want to ask. No one is having any question?
No, sir.
Fine. This picture we have seen 3-4 times. Roughly, you might have understand the basic themes of this replication process, how it is going to happen and what is going to happen. So these are the different components which we require when we are going to initiate or when we are going to do the DNA replication process. We require a helicase enzyme. Chemically, it is the product of DNA B protein. So it is called as DNA B protein or helicase enzyme. Primage, which is RNA polymerase, it is the product of DNA G gene. Remember, normally when we are writing genes and proteins, so this is the protein. When we are going to write the gene's name, that time I will write in italics. So it will write like this one, DNA B. So when we will write in italics, it means I'm talking about the gene. When it is the normal script, it is called as protein. So we require one helicase, we require a primase, and then we require different polymerase 1 and polymerase 3. This topoisomerase we require for removing supercoiling, we require ligase, and we require SSB protein, single stranded binding protein. These are the basic requirement for replication fork or replication processes. Now, yesterday also I have told you the mistakes. When DNA, we are going to make a DNA, the very little mistakes will be there. So roughly E. coli bacteria, which does not have so much sophisticated tools and techniques are there. So, means their systems are very primitive system. So they also doing one mistake in roughly by addition of 10 to the power 9 or 10 base pair. If they will add this much base pair, only one base pair wrong addition will be there. How they are going to maintain this much accuracy? Even if I will tell you to write this many alphabets, this many words, certainly you will do more than the bacteria as doing the mistakes. You will do more mistake than the bacteria. So how this correctness occurs? How this purity occurs? So this purity occurs at 3 level. First, show you the picture. So the first what we call it accuracy occurs at base selection process. When DNA polymerase going to select a nucleotide for the addition, so that base selection, base means nucleotide selection by DNA polymerase, they provide this much accuracy, 10 to the power 4 to minus this much accuracy is provided by the DNA polymerase at the base pair selection, complementary base selection. Then if they have selected a wrong base, then that will be repaired by proofreading capacity. Proofreading means seeing the added nucleotide, look at it is right or wrong. If it will be wrong, replace it. If it is right, then move forward. So that is called as proofreading. So this is proofreading capacity. So first level of purity, first level of what we call it mistake means accuracy is at base selection process, that is highest one. Then lowest means accuracy occurs by proofreading. Even by proofreading, if they have done some mistakes, they have done mistake here, they have done mistake here, then also they have a chance that they can repair by DNA repair mechanism. Like mutation, they are going to remove it. So there they can repair like this one. So overall, if you will add all three together, this much accuracy comes. So the accuracy in the DNA replication occurs by three different stages. The first highest accurate process is the DNA polymerase selection. So they select a correct nucleotide, correct complementary base pair that provides the highest accuracy. The second highest accuracy comes by DNA repair system, and the lowest accuracy comes from the proofreading power. Overall, all three together gives this much accuracy. So by adding this much nucleotides, they are going to do only one mistakes. Correct? Okay.
Yes, sir.
Okay. E. coli complete DNA, if you'll see, they are having only this much base pair, 4.6 multiplied by 10 to the power 6 base pair. It means one E. coli, if they are going to replicate themselves 1,000 times or 10,000 times, one nucleotide error will be there. To occur this one, if you will divide this one by this 10 to the power 9 divided by 4.6 multiplied by 10 to the power 6, so you will get 1,000 or 10,000. It means if bacteria will replicate 1,000 times, 1,000 generations, then one wrong nucleotide they will add it. So that much accuracy they are going to maintain, and that will be by three stages. First, DNA polymerase, this much, proofreading 10 to the power to the 10 to the power 3, and last one will be repair system, repair mechanism. Okay. Now we see the DNA polymerase, which type of enzyme bacteria is having, and what are their characteristic properties. So the first time DNA polymerase was discovered by a scientist in 1956, after 3 years after the DNA discovery. DNA structure was given by Watson and Crick, double-stranded DNA in 1953, and 3 years later, Arthur Kornberg discovered first DNA polymerase 1. Why it is 1? Because it is the first enzyme. So this was for this one they have got Nobel Prize also in 1959 in the physiology and medicine. Then later on they have discovered DNA polymerase 2, that was much later in 1970s by the same Kornberg only. So this is polymerase 1, polymerase 2, and polymerase 3 of bacteria. So, and these are the properties. So what I have, polymerase 1 is the product of Pol A gene. See here, Pol A, Pol B, Pol C. They are written in italics. It means I'm talking about the gene. Polymerase 1 have only one protein subunit. Only one protein part is there. Polymerase 2 have 7 proteins, and polymerase 3 is very complicated. More than 10 different proteins together comes and make a holo enzyme. If you have studied enzymes, we are going to call holo enzyme. Holo enzyme means, can anyone give me the definition of holo enzyme? Apoenzyme, coenzyme, and holo enzyme. Can anyone will tell me?
Sir, holoenzyme is apoenzyme plus coenzyme.
Okay. Okay. So, what is exactly holo enzyme? What to whom I am calling? What is the definition of holo enzyme? Because I have written apoenzyme and I have written coenzyme. So you are telling that if I will combine both, then it will be holo enzyme. But what is the exact definition of holo enzyme? Holo enzyme means a complete functional enzyme is called as holo enzyme. If you are having only proteinous part of enzyme, only one component which is non-functional. So it is half of the portion which is non-functional enzyme. If you are having only coenzyme, it is non-functional. When you will join these two, then it makes a functional enzyme, and that is called as holoenzyme. Similarly, in this case, polymerase 3, if we are having more than 10 different proteins, more than 10 different components, so if only one protein, if you'll take from this out of 10, it will be non-functional. If you'll take two protein, it will be non-functional. We require all 10 proteins together, assemble, and make a complete functional enzyme that we'll call as holoenzyme of polymerase 3. I will show you how it looks, it will be looking like. Now you see here, polymerase 1, polymerase 2, polymerase 3 all have polymerase capacity because they are polymerase, so certainly they will have a polymerase capacity. Proofreading power means 3,5 exonuclease activity. It is present one or in all the three cases. So proofreading, all can check their added nucleotide, it is right or it is wrong. Okay. 5,3 exonuclease because other enzymes does not require it. As you have seen, this 5,3 exonuclease polymerase 1 uses for the removal of RNA primer. So only RNA, this one polymerase 1 have this 5,3 exonuclease just for the removal of RNA primer. And yesterday I was talking about this capacities, polymerization rate, that has fidelity 16 to 20 base pair per second, 40 base pair per second, and 252,000 base pair per second. That what I have called yesterday fidelity or polymerization rate. Then I had talked about this processivity also, means one enzyme can add how many nucleotides. So if you'll say this polymerase 1 3 to 200 base pair to 1500 base pair, and this can add more than 5 lakh base pair. So this is the basic differences between the polymerase 1, polymerase 2, polymerase 3. Questions most of the time comes on this capacity, proofreading power, exonuclease. When we are going to talk about comparison, so then questions comes on this one. Okay, which enzyme is having proofreading capacity? Proofreading means it is 5,3 exonuclease or 3,5 exonuclease. So remember this one, this portion. Later on, polymerase 4 and polymerase 5 has been also discovered, and that was normally going to be used in DNA repair system. So total 5 in the DNA polymerase bacteria also have, but most commonly, most of the book contains these 3 only because we have detailed information about this one. So about polymerase 4 and 5, we don't have that much information. Clear?
Yes, sir.
Fine. When we are talking about this is the polymerase 1, 2, 3, 4, 5. If you'll see this one, polymerase 1, polymerase B, and this is polymerase, third polymerase. See how many components it is having. 10 to 15 base pair means nucleotide means protein components it needs, and so it have 2 alpha, 2 tau, epsilon, and every component is having their own significant. Later on, I will tell you what are their functions. We have a table. PPT I will send it to the sir's mail ID, and he will give it to you. There we are having function also. So this structure is very complicated. This is prokaryotic in the bacterial cells. In case of eukaryotic cells, our enzymes are more complicated than this one. So whatever information we are going to have, that is on the basis of bacterial study. So some information we need on polymerase 1, polymerase 2, and polymerase 3. Polymerase 1 encoded by polymerase Pol A gene, and it is ubiquitous among the prokaryotes. Ubiquitous means it is present in almost all the prokaryotic systems. Polymerase 1 normally used in the repair systems or small DNA fragments. So it is having 3,5 exonuclease, 5,3 exonuclease, and 5,3 polymerase activity also. Normally, it is going to synthesize lagging strand of DNA. Polymerase I is the most abundant polymerase, occurring more than 95% of the polymerase of bacteria is of type 1, polymerase I. This can add only 15 to 20 nucleotide per second, so it is having very poor processivity, very slow enzymes. Instead, polymerase I starts adding nucleotide to the RNA primer, because this is not only for polymerase I. Any DNA polymerase they require a RNA primer. So primer is required for all the DNA polymerases. Okay. Polymerase II, normally we are not using for replication process mainly. It is mainly used for DNA repair systems, and they have this 3,5 exonuclease, means proofreading power other than this polymerase activity. That is their significance. They are having little bit more processivity, higher speed than the polymerase I. This is the most important enzymes which we should understand in the detail. So polymerase III holoenzyme is the primary enzyme involved in DNA replication process, belongs to family C polymerase. This enzyme is having number of different components. We have seen more than 10 to 20 protein component it is having. One portion is called as protein 3 core. Second one is called as beta sliding clamp. Third portion is called as processivity factor, and one is called as clamp loading complex. Just I will show you the structures. These are the different components. Need not to remember everything. Some components are essential, need to be remembered. So alpha, it is having 2 in number, and they are for actual polymerization activity, means actual polymerase capacity nucleotide addition will be done by alpha component. So that you need to remember which protein component, protein subunit of DNA polymerase III is responsible for nucleotide addition, that is alpha. And how many alpha? 2 alpha. Then proofreading capacity, that will be done by epsilon. That also how many? 2. So and so this 2 and this whole A theta, these 3 together makes a core molecule. We have seen there are many components. So one is core enzyme. So 3 components makes core enzyme. If you'll see the structure here, core polymerase is alpha, epsilon, and theta. Alpha for polymerization, epsilon for proofreading, and this does not have function, is not defined. Then we are going to see the clamp loading complex. This is the nature of clamp loading complex. Tau in 2 in number, gamma and delta, delta dash. They are called as clamp loader. Clamp means beta is clamp and this is clamp loader, means it is holding the DNA strands. This is the rough cartoonic diagram of polymerase. So you can see here, this is not the part of DNA polymerase, this is the helicase. This is core component. This is alpha, epsilon, theta. So one core here and one core here. So 2 core enzyme it is having. This white portion here you can see, that is the beta clamp. And to load this beta clamp, we are having a clamp loader. So this component is called as clamp loader. So 2 core, 1 clamp loader, and 2 clamps. This is the normal structure of polymerase III in case of bacteria or E. coli. So you need to remember the function of first 2, alpha and epsilon. Rest other are clamp, loader, and clamps only. Just wait for a second. I have a video, so let me show you the video how it works, and then we'll go detail.
Cells, like these prokaryotic E. coli cells, replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of four nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established.
Actually, here you will see, sorry. Just 1 second.
Cells like these prokaryotic E. coli cells replicate themselves quickly and efficiently. Part of the process of
Actually,
asexual reproduction.
there you have seen the one how DNA polymerase works. So that I was planning to show it to you. Just forgot.
Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of 2 strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make two new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication.
So this is a one replication bubble where you can see this is one replication fork this side and another replication fork this side. So from both sides we are having replication is going to occur. So this is one replication fork we are talking, only half of the Y shape structure we are talking, and this is one DNA polymerase. This is helicase, just now you have seen it. This is clamp loader, and here we are having clamp, and this is the core enzyme. So on this strands, if you'll see the mouse, so on this strands DNA is going to move in this direction. So this is 5' to 3' direction, and on this another opposite strand, on this is lagging strand. So you have seen this is one strand has been synthesized here, and opposite strand synthesis has been started from here. So this will go up to here, and then this DNA polymerase one will come and it will remove this yellow portion which is RNA primer and then replace with the DNA.
Understand how complex eukaryotic organisms replicate DNA.
So you can see here in this case, this is continuously growing on, but on this strand, this DNA is single stranded because it has been started from here. So again, for this region, it needs another small DNA need to be formed. That is called as another Okazaki fragment.
Scientists first studied
So see here.
replication in prokaryotic
Again,
models.
clamp loader came, RNA polymerase primer forms. This is primer. This is DNA polymerase, which has removed the primer and added it, joined it with the help of replace it. So this is and this is Okazaki fragment is going to form. So clamp loader just loading the clamps and it is continually going on.
Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize.
So this is continuously going on till and here you can see if it is going to unwind this DNA, ultimately there will be a supercoiling in the further region, and that will be removed by this gyrase. So gyrase, what it is going to do, we are having different types of gyrase enzyme. They cut one of the strands or both the strands and then rotate it so that ultimately supercoiling will be removed. Like you can see here.
Double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme Primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, This process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides.
And then ligase will come and it will join it.
Ligase is used to ensure bonding between the fragments and
Is it clear for everyone?
Yes, sir. Yes,
the
sir.
replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell. Cells.
Okay. So this is how it is going to occur. This is how new nucleotide will be added at 3' end. Sorry. So how a new nucleotide comes in the triphosphate forms, 3 phosphate, like adenine, adenosine triphosphate, guanosine triphosphate, cytosine triphosphate. This is the 3' OH, which is the growing chain. So when a nucleotide comes, a new nucleotide comes, it comes in the triphosphate forms. From these triphosphate molecules, 2 phosphate. This phosphate we are writing it like AMP. Then we are having 1 phosphate, and then we are having another phosphate. This is called as alpha phosphate. This is called as beta phosphate. This is called as gamma phosphate. So beta and gamma phosphate will be removed, detached in the form of pyrophosphate, and alpha phosphate is associated with the A in the form of nucleotide monophosphate, and that will be joined here with the help of polymerase enzyme. This is the polymerase enzyme, which looks like a closed hand, means palm like a structure. So this new nucleotide will be added in the form of 1 phosphate. So 1 phosphate from this new nucleotide form a bond with this OH after the removal of pyrophosphate in the 5'. Again here OH is free. Again a new nucleotide will come with triple phosphate, triphosphate. 2 phosphate will be removed and 1 phosphate will be added. So this is continuously growing on the chain. This is like this one. This is a nucleotide because here we are having A, so obviously we require thymine, and thymine comes in triphosphate form. This is alpha, beta, gamma. So from this one, this beta gamma will be detached in the form of pyrophosphate, and this molecule will join here in the form of nucleotide. So this is 1 nucleotide has been added. Again this process will continue. So this is how your chain grows. And proofreading, I have already told how it occurs. So proofreading means the correction of wrong nucleotide. This is 10 to the power 10 base pair correctness, and that is coming from the 3. This proofreading capacity, like this is your DNA polymerase 1, polymerase 2, polymerase 3, because all the enzymes have proofreading capacity. So one component, one portion of the DNA protein works as the polymerase power, and one portion works as the proofreading capacity, because both are two enzymatic activity. This is exonuclease activity and this is polymerase activity. So one protein is having two activity. This is called as bifunctional enzyme. Bi means two functions on the same protein at the same time are called as bifunctional enzyme, by which it is going to do the detection and removal of the wrong nucleotide. This is DNA. This is your RNA portion, RNA fragment, RNA primer. For time being, this DNA polymerase, which is growing in this direction. So on this DNA polymerase is there, which will remove this nucleotide RNA portions and then add 1 nucleotide. So DNA polymerase removes this 5'3" exonuclease activity, will remove 1 nucleotide. So this nucleotide has been removed, and then add 1 nucleotide. So it is DNA will grow up to here. And then the gap between 2 nucleotides will be sealed by DNA ligase. So this is how your DNA is going to be synthesized. One thing which I left till here is initiation part, because that is the one of the most important part. Continuously the chain reaction means So this is that initiation I will do after this ligation only. This ligation is the joining of when you are going to join two nucleotides. Means if you'll see here, we are having one nucleotide this side, which is having 3' OH, and we are having one DNA portion this side, which is having a phosphate. The gap is there. So this gap need to be filled by joined together by forming a phosphodiester bond. You know that two nucleotides joined together by a phosphodiester bond. So the ligation means the formation of phosphodiester bond between one nucleotides 3' OH and another nucleotides 5' phosphate. So remember, 3' hydroxyl at one end of the DNA, 5' phosphate at the another end of the DNA. There is a ligation reaction occurs between these two components. In this ligation reaction occurs in three different stages and two different types of ligase enzyme we are having. One kind of enzymes which uses ATP as a source substrate, and another is which uses NAD molecule. Remember, write down in your notebook. The DNA ligase which is going to be purified from bacteria, they use NAD molecule, nicotinamide adenine dinucleotide. And the DNA ligase which we are going to purify from virus and eukaryotic cells, they uses ATP. Where they use ATP and where they use AMP. So see here, this is the reaction, ligation reaction. One by one, forgot about this portion. Just see only those portion which I am going to tell you. So this is my one reaction, first step reaction. This is my second step reaction, and this is my third step reaction. So we are having ligation. Ligase is an enzyme. Enzymes means it is a protein. So ligase is having a enzyme that amino acid that is called as lysine. We have seen lysine previously also in case of histones also. This lysine enzyme is a basic amino acid, so it is having one extra amino group. Clear? Now I have told you either I am going to use ATP or I am going to NAD. Both contains. This is also going to contain AMP fragment, adenosine monophosphate, and this also contains AMP fragment. So from both the things I am going to take AMP, adenosine monophosphate. Either the substrate is ATP or NAD, we require AMP. So AMP will be taken. This is your AMP, adenosine monophosphate, adenosine ribose and phosphate. In the first step of this ligation reaction, this AMP portion is going to join with this enzyme, lysine of the ligase enzyme. So you can see this is your enzyme and AMP has been added here. So this is called as adenylation of the enzyme. This is my first reaction. If you are using bacterial DNA ligase, then we will use this as a substrate AMP source. If you will take viral DNA ligase, then you will use ATP as a source, ATP as a AMP source. Clear? First step is clear?
Yes, sir.
Okay. In the second step, this AMP portion from the enzyme need to be transferred this phosphate. This is I am going to join these two molecule. This hydrogen OH and this phosphate need to be joined. So this phosphate will take this AMP portion. So in the second step, the phosphate at the nick means gap, will be activated. So this AMP you can see here, this is the AMP molecule which has been transferred to this phosphate, and enzyme is free. And in the third step, the lone pair of OH attacks the phosphate, removes this AMP portions which came from the enzyme, and bond is going to form. So if you don't want to remember complete process, remember only few things. For sealing of two different nucleotide, two fragment of DNA need to be joined together by a enzyme that is called as DNA ligase. Which amino acid of enzyme participate? It is the lysine amino acid of enzyme which participate. it requires a AMP fragment which comes from either ATP or NAD. In case of bacteria, NAD is the source. In case of virus and eukaryotes, ATP is the source. This much minimum you need to remember. If you remember complete process, it is perfectly fine. If you don't want to remember complete reaction, minimum remember these three things. Any question? Any question? Anyone didn't understand? Anyone is having anything, you can ask. If you have not understand also, you can ask me, I will repeat it.
It's clear, sir.
For everyone or only for you?
Yes, sir.
Saurav, you are telling me on behalf of everyone, right?
Yes, sir. Clear, sir.
Okay. Termination we will do later on. Leave it termination, because termination and regulations we will do later. Just I am going to tell you the initiation. Just wait for a second. Initiation, I think I have escaped it. Okay. This portion let me finish first. Initiation. What I've told you, bacterial DNA replication initiates at a specific place. That portion is called as origin of replication. Okay. This origin of replication is composed of 245 base pair. So bacterial origin of replication consists of 245 base pair. In this 245 base pair, there are 7 specific regions. Like in case of this is a bacterial DNA, which is single circular DNA. So this portion is origin of replication. In this origin of replication, this is 245 base pair. This 245 base pair contains 7 specific regions. Means it contains 13 base pair sequences here. Means it contains 13 base pair sequence. So 3 multiplied by 13 base pair. And 4 9 base pair sequences. 1, 2, 3, 4. So 4 9 base pair sequences. Means bacterial origin of replication is composed of 245 base pair. Just for example, this is the origin of replication, which is of 245 base pair. Inside this 245 base pair, bacteria is having 13 base pair repeated sequences and 4 9 base pair repeated sequences. Most of the time, this 9 base pair sequence we are going to call DNA A box. Okay. So at least 9 different enzymes or protein participate in this one. In the first step, 4 to 5 DNA A protein. अब आप you should remember, आपको याद होना चाहिए DNA A protein मैंने किसे बोला था? DNA A protein. This is the DNA A protein which recognizes the origin of replication. तो पहला प्रोटीन जो origin of replication को recognize करता है उसको बोलते हैं DNA A protein. So it requires 4 से लेके 5 DNA A protein comes and binds to this 9 base pair sequences. जैसे आप यहाँ देखेंगे, this is a sequence here. So this is DNA A protein which comes along with ATP. हर एक DNA A protein के साथ एक ATP associated होता है. So this DNA A protein along with ATP comes and binds with this 9 base pair sequences and make a loop-like structure. This is the first step. समझ में आया? 245 बेस पेयर सीक्वेंस का ओरिजिन ऑफ रेप्लिकेशन होगा, जिसके अंदर में हमारे पास 7 स्पेसिफिक रीजन्स होते हैं। 13 बेस पेयर का 3 रीजन, 9 बेस पेयर का 4 रीजन्स। डीएनए ए प्रोटीन सबसे पहले आएगा एटीपी के साथ, और दैट विल बाइंड्स विद दिस 9 बेस पेयर सीक्वेंस एंड मेक ए लूप लाइक स्ट्रक्चर। ठीक है, एंड इन दिस केस ये 13 बेस पेयर रीजन्स जो होता है, अगर आप देखेंगे इसका सीक्वेंस, तो दैट इज 80 रिच रीजन। ये एक सीक्वेंस की बात कर रहे हैं। टी के कॉम्प्लीमेंट्री में एडेनिन होगा, सो इसके पास बहुत सारे एडेनिन होते हैं, एडेनिन एंड थायमिन होते हैं। सो 13 बेस पेयर सीक्वेंस इज वेरी 80 रिच रीजन। तो जब ये बाइंड करते हैं, पहला सबसे पहले डीएनए ए प्रोटीन एटीपी के साथ आकर बाइंड किया, उसके बाद एक दूसरा प्रोटीन आता है जो हिस्टोन लाइक प्रोटीन होता है, जिसको बोलते हैं एचयू प्रोटीन। ये भी एटीपी के साथ आता है, और ये आके बाइंड करता है यहां पे, ये स्टार्ट करता है डीएनए अनविंडिंग। ठीक है, उसके बाद फाइनली आता है डीएनए हेलिकेज। जो डीएनए बी है दैट इज हेलिकेज। 6 डीएनए ए मिलके एक हेलिकेज बनाता है कंप्लीट, जो आप यहां देख रहे हैं। 6 डीएनए बी प्रोटीन मिलके एक हेलिकेज बनाता है इस तरह का, जो डीएनए को फाइनली 2 हेलिकेज बाइंड करेगा, एक फॉर्क पे, दूसरा दूसरे फॉर्क पे, एंड it will move in this direction, it will move in this direction. This is how DNA initiation starts, DNA replication starts. इतना समझ में आया?
Yes, sir. Yes, sir.
So what I have told, this is the explanation of those whole processes. Initially DNA A tetramers recognizes and binds to the 4-9 base pair repeats, जिसको हम DNA A box भी बोलते हैं, at the origin of replication, और इसे additional molecule of DNA A binds cooperatively and finds a form a closed complex, जिसको मैंने बोला loop like structure बनाता है। उसके बाद HU प्रोटीन आता है along with DNA means ATP and start opening the 13 base pair regions, which is 80 rich region. Fine. So a 13 base pair region is 80 rich region, जिसको ओपन करता है, जहां से डीएनए इनिशिएशन स्टार्ट होता है। सो इस जब HU प्रोटीन इनिशिएट करता है, उसके बाद आता है डीएनए बी एंड डीएनए सी। डीएनए सी इज एक्चुअली हेलिकेज एंजाइम, जो 6 मॉलिक्यूल मिलके एक हेलिकेज बनाता है, और डीएनए सी इज कॉल्ड एज हेलिकेज लोडर। वो बी को बाइंड करने में हेल्प करता है। तो अल्टीमेटली वी आर हैविंग ए 6 बेस पेयर 6 एंजाइम हेलिकेज लोडेड हियर, एंड दैट इज गोइंग टू अनविंड योर डीएनए। जब आपका डीएनए अनविंड हो गया, देन कम्स प्राइमेज एंजाइम। इट बाइंड्स हियर एंड मेक्स ए स्मॉल प्राइमर मोलिक्यूल्स, एंड उसके बाद डीएनए पॉलीमरेज 1 एंड पॉलीमरेज 3 विल कम। ठीक है, this was roughly DNA polymerase, means how replication initiates and elongates. अब डीएनए रेप्लिकेशन हमेशा नहीं होता है। डीएनए रेप्लिकेशन आपका, जब हमने डीएनए को रेप्लिकेट कर लिया, मान लीजिए दिस इज ए डीएनए, इट इज गोइंग टू बी रेप्लिकेटेड, ये 2 कॉपी बना दिया, इंस्टेंटली दिस डीएनए विल नॉट गो फॉर अगेन रेप्लिकेशन, तुरंत नहीं जाएगा रेप्लिकेशन में। इट नीड सम पॉज, इट नीड सम टाइम। दैट टाइम लैप्स, जो ये डीएनए को टाइम चाहिए होता, दैट प्रोसेस इज कॉल्ड एज डीएनए सीक्वेस्टरिंग। एंड इस सीक्वेस्टरिंग में मिथाइलेशन का रोल होता है। ठीक है, ये इतना समझ में आया? मीन्स रेप्लिकेशन खत्म हुआ, दूसरा रेप्लिकेशन, दूसरा साइकिल तुरंत स्टार्ट नहीं होता है। 2 रेप्लिकेशन के बीच में थोड़ा सा टाइम लैप्स चाहिए होता है, पॉज चाहिए होता है। उस टाइम पॉज को कैसे देते हैं सेल को या डीएनए को कैसे देते हैं? थ्रू ए प्रोसेस दैट इज कॉल्ड एज सेक्वेस्टरिंग ऑफ डीएनए। इस सेक्वेस्टरिंग ऑफ डीएनए में मिथाइलेशन का एक रोल होता है। ठीक है, डैम, और वो मिथाइलेशन कौन करता है? डैम मिथाइलेशन, जो मैंने पहले भी देखा था। इसको ये मैं बता के फिर छोड़ देता हूं आज के लिए। आपके पास एक डीएनए है, इस डीएनए में हमारे पास नॉर्मली ये सीक्वेंस होता है GATC। अगर इसको हम इस तरफ यहां लिखें GATC, ठीक है, दिस इज ए डीएनए विच इज GATC। इसमें एडिनिन जो मॉलिक्यूल है ये मिथाइलेटेड होता है, दिस इज मिथाइलेटेड। इसको मैं स्टार मार्क कर देता हूं, स्टार मतलब इट इज मिथाइलेटेड, ठीक है। अब दिस डीएनए इज गोइंग फॉर रेप्लिकेशन, तो ये हमारा एक डीएनए मॉलिक्यूल होगा, ये एक दूसरा डीएनए मॉलिक्यूल होगा GATC, एडेनिन इज मिथाइलेटेड, GATC, एडेनिन इज मिथाइलेटेड। सो दिस इज वन टेम्पलेट एंड दिस इज अनदर टेम्पलेट। अब मैं इसको दूसरे कलर से लिखता हूं। This is a newly strand which is formed. Here we are having GATC and here we are having GATC. In this case, this DNA, both the adenine, both the strands are methylated. So this is completely methylated DNA. जब रेप्लिकेशन हुआ तो एक स्ट्रैंड जो पेरेंटल स्ट्रैंड है दैट इज मिथाइलेटेड एंड जो नया स्ट्रैंड फॉर्म है दैट इज अनमिथाइलेटेड। So this is called as hemimethylated DNA. Hemi means half. This is also hemimethylated DNA. This is also hemimethylated DNA. So जब रेप्लिकेशन होता है तो हमारा डीएनए बैक्टीरिया का डीएनए कंप्लीटली मिथाइलेटेड नहीं होता, दे आर हेमीमिथाइलेटेड। सो दिस हेमीमिथाइलेटेड डीएनए गोज एंड बाइंड्स विद द प्लाज्मा मेम्ब्रेन फॉर सम टाइम। रेप्लिकेशन हो गया यहाँ, after replication this DNA, hemimethylated DNA will go and associated with the plasma membrane for some time. इस प्रोसेस को, डीएनए को प्लाज्मा मेम्ब्रेन में जाके एसोसिएट होने के प्रोसेस को हम बोलते हैं सेक्वेस्टरिंग ऑफ डीएनए। once this sequestering of DNA, after some time this DNA will come back and then this adenine will be again methylated. So जो hemimethylated DNA sequestering के लिए गया था वो आएगा and that will be completely methylated by डैम मिथाइलेज एंजाइम। सो डीएनए का वो टाइम जब वो हाफ मिथाइलेटेड हाफ अनमिथाइलेटेड स्ट्रैंड में था, दैट इज गोइंग फॉर सेक्वेस्टरिंग प्रोसेस। और जब सेक्वेस्टरिंग होता है दैट टाइम डीएनए डज नॉट गो फॉर रेप्लिकेशन प्रोसेस। सो फॉर रेप्लिकेशन डीएनए शुड बी कंप्लीटली मिथाइलेटेड। इसीलिए इस स्टेज में डीएनए रेप्लिकेशन नहीं होता, बंद होता है जब तक वो कंप्लीटली मिथाइलेटेड ना हो जाए। क्लियर?
सेल डिवीजन से पहले होगा ना मिथाइलेशन?
सेल डिवीजन से पहले, डीएनए रेप्लिकेशन के जस्ट बाद। मतलब आपका एक सेल साइकिल है जिसमें हमारे पास इंटरफेज होते हैं। इंटरफेज में मैंने डीएनए रेप्लिकेशन कर लिया, ठीक है। इंटरफेज में डीएनए रेप्लिकेशन हो गया, राइट? हेलो?
यस सर।
आफ्टर रेप्लिकेशन this cell has to go for mitotic phase or meiotic phase, that is prophase, anaphase, metaphase, telophase. उसी प्रोसेस में means after interphase, once DNA has been duplicated, again this DNA will not go for duplication till cell is going to completely divide. So ये सेल डिवीजन के बीच में ही होता है, दो इंटरफेज के बीच में होता है सेक्वेस्टरिंग।
ओके सर।
ओके, this is clear for everyone?
Sir, excuse me.
Yes, yes.
सर कितने सेकंड से कितने मिनट्स का टाइम लैप होता है?
नहीं, ये तो मिली माइक्रो सेकंड्स में होता है, सेकंड्स मिनट्स नहीं होगा। 20 मिनट का तो टोटल रेप्लिकेशन साइकिल ही होता है बैक्टीरिया का, जिसमें उनको इंटरफेज भी कंप्लीट करना है, माइटोटिक मियोटिक फेज भी कंप्लीट करना है, सारी चीज करना है 20 मिनट में ही नॉर्मली होते हैं, इसीलिए मिली माइक्रो सेकंड्स में होते हैं। और ये GATC की जो मैं बात कर रहा हूँ ये होता है उस विदिन 245 बेस पेयर के सीक्वेंस में ही, इसी 245 न्यूक्लियोटाइड का जो ओरिजिन ऑफ रेप्लिकेशन होता है उसी के बीच में होता है ये GATC सीक्वेंस। रफली उस 245 बेस पेयर में 11 से ज्यादा जगह पे ये GATC इस सीक्वेंस में होता है, 5 प्राइम पे G एंड 3 प्राइम पे C। आप यहाँ देख रहे हैं। So it has roughly 11 of them in the 245 base pairs. सो उसी का एक्सप्लेनेशन लिखा हुआ जो मैंने आपको अभी बताया, उसी का रेप्लिकेशन है. इमीडिएटली आफ्टर द रेप्लिकेशन द डीएनए इज हेमी मिथाइलेटेड, द पैरेंटल स्ट्रैंड हैव मिथाइलेटेड and the newly synthesized strand does not have any methyl group, so it is called as hemi-methylated, and they go for sequestered for a where they are going to interact with the plasma membrane. This is inactive phase, you can call it non-replicative phase. ठीक है. तो यहां तक इज इट क्लियर फॉर एवरीवन? ठीक है. सो डीएनए रेप्लिकेशन आपको प्रॉब्लम, आई डोंट नो व्हाट इज द इशू, या तो मैं थोड़ा ज़्यादा बता रहा हूँ बिकॉज मुझे सर ने बताया था कि ये दिस इज फॉर CSIR, तो जिस तरह से क्वेश्चन आते हैं मैं उसी तरह से आपको बता रहा हूँ. अगर फिर भी लग रहा है कि मैं फास्ट हूँ या मैं मतलब उनको प्रॉब्लम है तो मुझे बताएंगे, तभी मैं समझ में आएगा मुझे. ठीक है. हेलो.
यस सर.
हाँ, तो आपके जो कलीग्स हैं प्रोबेबली यू माइट हैव सम ग्रुप्स, व्हाट्सएप ग्रुप्स एंड अदर थिंग्स, राइट? तो आप वहाँ एक बार बात करके मुझे बताएंगे, या जहाँ बता सकते हैं, या तो फिर सर को बता दीजिए, सर मुझे कम्युनिकेट कर देंगे, या फिर मैं अपना नंबर दे देता हूँ, मुझे आप कम्युनिकेट कर दीजिए कि स्टूडेंट को कहाँ दिक्कत हो रही है, मतलब वेयर दे आर फेसिंग प्रॉब्लम. या तो कंटेंट ज़्यादा लग रहा होगा, तो चूंकि CSIR में आपको नॉर्मल MSc से पोस्ट ग्रेजुएशन से थोड़ी ज़्यादा के क्वेश्चन आते हैं. जो आपने MSc में पढ़ा होगा उससे थोड़ा ज़्यादा क्वेश्चन आएगा. ठीक है. तो इसीलिए मैं उतना ही को देने की कोशिश कर रहा हूँ जितना CSIR में आता है, जो प्रीवियस ईयर्स के क्वेश्चंस में आते हैं. अगर ये ज़्यादा लग रहा है फिर तो जो बताएंगे तभी मेरे पास सॉल्यूशन होगा.
सर इफ यू कैन इंक्लूड सम प्रीवियस ईयर क्वेश्चंस.
या वो मैं आपको देता हूँ. मैंने बोला था कि चूंकि मैं क्वेश्चन अभी इसीलिए नहीं दे रहा था क्योंकि मैंने सर को बोला था कि मैं आई विल कंडक्ट ए टेस्ट, आई विल गिव यू 15-20 क्वेश्चंस, वो आप सॉल्यूशन करेंगे तो आपको पता भी चलेगा कि आपने कितना पढ़ा है, कितना समझा है. तो इसीलिए मैं क्लास में नहीं बता रहा था, मैं दे देता हूँ. मैं कल क्वेश्चन लेके आऊँगा कुछ.
ओके सर.
ये मेरा नंबर है. आप स्टूडेंट, आप किसी आपका बैच में से कोई भी मुझे शेयर कर देंगे कि स्टूडेंट को कहाँ दिक्कत आ रही है. ठीक है. सो दैट आई विल अंडरस्टैंड कि उनको कंटेंट में दिक्कत है या नहीं समझ पा रहे हैं या क्या इशू है, मुझे पता नहीं. जब तक बताएंगे तब तक मुझे समझ में नहीं आएगी. ठीक है. एनी क्वेश्चन? आप में से किसी के पास कोई क्वेश्चन हो तो बताइए.
सर
सर
सर डीएनए रेप्लिकेशन बाइडायरेक्शनल होता है, तो जैसे एक डायरेक्शन में जो स्ट्रैंड लैगिंग स्ट्रैंड है वही दूसरे डायरेक्शन में लीडिंग स्ट्रैंड होगा ना?
मैं दिखाता हूँ. जैसे ये एक फॉर्क है, ठीक है. तो इसको मैं दो पार्ट में डिवाइड कर लेता हूँ. ठीक है. मान लीजिए ये यहाँ ये लीडिंग बना रहा है, यहाँ ये लैगिंग है. ठीक है. जब हम फॉर्क की बात करते हैं, व्हेन वी आर टॉकिंग अबाउट हाफ ऑफ द फॉर्क, तब हमें इस तरह से दिखेगा. इसी में इस स्ट्रैंड पे ये कंटिन्यूअसली बनता जाएगा और यहाँ ये हाफ बनते जाएंगे, इस तरह से बनते हैं. ठीक है. तो नॉर्मली हम इतना देखते हैं, इसीलिए मुझे लगता है कि एक लीडिंग है एक लैगिंग है. बट अगर आप पूरे स्ट्रैंड्स को, पूरे बबल को देखेंगे तो आधे के बबल पे लीडिंग चलता रहता है, आधे बबल पे लैगिंग चलता रहता है हर एक स्ट्रैंड्स पे.
ओके सर.
एनीवन एल्स? ठीक है. मैंने नंबर दे दिया आपको. अगर आप कुछ शेयर करना चाहें, कोई प्रॉब्लम हो, जहाँ दिक्कत हो मुझे बताइए, तो आई विल ट्राई टू रिजॉल्व इट. ओके
सर
बताइए.
सर जो ओकाजाकी फ्रैगमेंट है, लीलो फ्रैगमेंट में डिफरेंस मतलब होता क्या है? क्योंकि उसमें समझ नहीं आता, कभी-कभी मतलब एक दूसरे को वो कर जाते हैं,
नहीं,
interrupt करते हैं।
एक स्ट्रैंड्स पे आपका डीएनए जो होता है वो कंटिन्यूअसली, जैसे ये यहाँ तक ओपन हुआ है डीएनए फ्रैगमेंट, डीएनए अनविंड जो जिप बना है ना, वो यहाँ तक बना है, तो ये स्ट्रैंड्स यहाँ तक, ये लीडिंग कंटिन्यूअसली बनता है इस डायरेक्शन में। आप डीएनए को अब यहाँ तक अनविंड करेंगे तो ये कंटिन्यूअसली इस स्ट्रैंड पे बनता जाता है, जबकि दूसरे स्ट्रैंड्स पे ना वो फ्रैगमेंट में बनता है, छोटे छोटे टुकड़ा में बनता है। इस छोटे टुकड़े को बोलते हैं ओकाजाकी फ्रैगमेंट। तो जब आप डीएनए को देखेंगे तो आप अगर हाफ पोर्शन को देखेंगे, हम अगर आधे ही रेप्लिकेशन फॉर्क को देखें तो एक पे हमें कंटिन्यूअस डीएनए बनता हुआ दिखता है, दूसरे पे हमें छोटे छोटे टुकड़े में बनता हुआ दिखता है। तो इसको बोलते हैं लीडिंग स्ट्रैंड्स, इस डीएनए को बोलते हैं लीडिंग स्ट्रैंड्स और कंटिन्यूअस स्ट्रैंड्स। दूसरे को जहां छोटे छोटे टुकड़े में बनते हुए दिखते हैं उसको बोलते हैं लैगिंग स्ट्रैंड्स और डिसकंटिन्यूअस स्ट्रैंड। ये लीडिंग और लैगिंग इसीलिए सिर्फ होता है ताकि 5 प्राइम टू 3 प्राइम डायरेक्शन को मेंटेन करके रखा जाए। अगर ये मेंटेन करने की प्रॉब्लम ना आती ना तो यहां भी कंटिन्यूअस ही बनता। ठीक है, यही क्वेश्चन था। क्लिनिक फ्रैगमेंट में बता रहा हूं, हमने बात किया डीएनए पॉलीमरेज 1 की। आपने देखा डीएनए पॉलीमरेज 1 हैड 3 प्रॉपर्टीज। एक प्रॉपर्टीज था 5 प्राइम टू 3 प्राइम पॉलीमरेज, जो सभी पॉलीमरेज के पास होता है। दूसरा था 3 प्राइम टू 5 प्राइम एक्सोन्यूक्लिएज, जिसको हमने बोला था प्रूफ रीडिंग। ठीक है, और तीसरा था 5 प्राइम टू 3 प्राइम एक्सोन्यूक्लिएज, जो प्राइमर को रिमूव करने के लिए चाहिए होता है। तो ये डीएनए पॉलीमरेज 1 की है। इस पॉलीमरेज 1 में से अगर मैं इस प्रॉपर्टीज को हटा देता हूं 5 प्राइम टू 3 प्राइम एक्सोन्यूक्लिएज को, तो वो डीएनए पॉलीमरेज 1 जिसके पास पॉलीमरेज एक्टिविटी हो और प्रूफ रीडिंग पावर हो, ये पॉलीमरेज कैपेसिटी मैंने निकाल दिया, उसको मैं बोलता हूं क्लीनिव फ्रैगमेंट। इस क्लीनिव फ्रैगमेंट का नॉर्मली हम यूज कहां करते हैं? हम यूज करते हैं जेनेटिक इंजीनियरिंग में, क्लोनिंग के टाइम पे।
क्लियर सर।
फाइन, और किसी के पास कोई क्वेश्चन हो? फाइन, ठीक है, आप एक बार पढ़िए। आप में से कोई अपना एक मेल आईडी मुझे, या सर को मैं अभी मेल कर देता हूँ, दोनों स्लाइड्स बेसिक्स का और ये भी सर आपको फॉरवर्ड कर देंगे। ठीक है, तो आप एक बार देख लीजिए, जहाँ दिक्कत लगे वहाँ मुझे बताइए, फिर मैं उसका सॉल्यूशन निकालूंगा। ओके।